Hello and welcome to your Chapter 9 video lecture where we are going to talk about infections. We'll start off discussing our microorganisms and humans. What's the relationship there? There are, of course, many different types of relationships. The first type we're going to talk about is symbiosis, and I'm not sure why this doubled up here, but this is a type of interaction between two different organisms that are living in a close physical association. So in this case, both organisms are going to have an advantage. So an example I put here is our clownfish that is going to feed on small invertebrates that otherwise have potential harm to the sea anemone. And what happens is the fecal matter from the clownfish will also be an advantage. It's going to provide nutrients to the sea anemone. The other type of relationship is the mutualism, and this is going to be when we have an interaction between two or more species where each species has a net benefit. So the example I'm giving here is the hummingbird drinking from a flower, and pollination is going to take place. So here the bird is getting the nectar, where the pollen is also going on to the hummingbird that can then go to other areas and release that pollen. And then we've got this idea of mutualism taking place within our human body too. Our gut flora is essential for efficient digestion. It benefits us, and we're also providing a home for all of the bacteria, fungi that live within us. Then we've got commensalism, and you could see this pictured over here, where we have this interaction where one of the species is going to benefit while the other one doesn't. It basically stays neutral. So an example of this is going to be this fish that is called the golden jackal. It follows the tiger shark around, and it's going to eat any leftovers from the tiger's meals. The next term we have is pathogenicity, and we talked about this in microbiology. This is going to help benefit the microorganisms, but unfortunately it harms the human. So essentially it is attacking our body in order to gain nutrients or residency and so forth. Next term is opportunism, and this is a situation where we're going to have benign microorganisms become pathogenic because of decreased human host resistance or translocation that takes place, taking the microorganism from one area of the body to another body site that it does not typically live within. And so I just wanted to also put this image over here discussing the normal microbiome, where we are going to have our microbes, again, hopefully in a mutualism state. But if we, let's say, aren't able to keep these microbes in check, they can lead to infection and eventually into disease. And in order to do this, the pathogenic microorganism has to be able to go through something called colonization, and that is their ability to survive and multiply on or within our human environment. Now let's talk about the processes of infection. So there are many different ways in which we can encounter, come into contact with a microorganism. We have these endogenous microorganisms that are present within our body and, like we said, a part of our normal microbiome. But sometimes they get a little out of control and can start causing issues. Our exogenous microorganisms are transmitted from an external source in our environment. It could be through our contaminated water, or maybe another person sneezes a virus out and we somehow bring it into our body. It could be through a mosquito bite or through our food in which these microorganisms come into our body. And then that process is transmission. It could be either direct or indirect. And by direct, we are talking about vertical transmission, meaning it's coming from the mother to a child through the placenta. We talked about how HIV in chapter 8 can move across the placenta to the fetus. But of course, there's other microorganisms as well, such as Listeria monocytogenes. And they can also be transmitted through breast milk in this manner. The other type is going to be through horizontal transmission, and this is from one person to another through exposure by blood and other body fluids. We can also have this take place through zoonotic infections. Like we said, it could be from an animal or an insect. Then we've got indirect transmission. At this point, we are using some other, let's say, inanimate object or through the air for it to come into our body. So if we have a table that somebody, wiped their nose with a napkin and then, I don't know, wiped it on the table, and then we touch that table and it infects our body. It could be through inhalation or a droplet infection. So if somebody has a common cold or pneumonia and passes it on to us. Ingestion of contaminated food or water would be good examples. And lastly, we've talked about colonization. How is that microbe going to survive and multiply within our body to continue infecting? We have the term invasion or penetration. This is going to involve the dissemination of the pathogen throughout our local tissues or our body, and the pathogens can produce exoenzymes or toxins to make this possible. I have an example of H. pylori, which is a bacterium here that's invading the lining of the stomach. You may remember this example from micro, where the microbe is again making contact with our stomach. So here's that mucin gel layer before it reaches the epithelial cell. And so it is able to hinder the bacterium from entering that epithelial cell by having this mucin layer. But if the bacterium, which H. pylori has, releases urease, which is an enzyme that is able to neutralize the stomach acid and break through this mucin layer and eventually make it over to the epithelial cell, that can then cause ulceration to take place. The next step is dissemination, to spread that infection. And this can happen by direct extension through surrounding tissues or within our cardiovascular or lymphatic vessels. So travel within our blood or our lymph within our lymphatic vessels. And then we've got our cellular or tissue damage, and this could be directly by production of toxins. So I put in this image here. Again, you may remember this from micro. When we have gram-negative bacteria, they have this extra area on top known as LPS. Do you remember that? Lipopolysaccharides. That is an endotoxin. And so this molecule that is found on their outer membrane is going to be responsible for toxic properties that can trigger the immune system's inflammatory response. And if we get a high concentration of the endotoxin here in our blood, it could drop our blood pressure and cause us to go into multi-organ failure and lead to death. Now, what are the factors for infection? The first term we need to know about is communicability, and this is the ability of that microbe to spread from one individual to others and cause disease. So you can see that we're going to talk about the stages of an infection in a couple slides, but we have this incubation period in which the microbe enters our body but hasn't really shown or made come about symptoms and signs of the disease. But once we start seeing those, it increases our ability to make it more communicable. And examples of this would be measles or pertussis that would spread really easily, whereas HIV has lower communicability because it is more difficult to pass that over to somebody. We can also kind of think of this as the Swiss cheese model for infectious disease. And I'm going to refer to this term here, infectability. It's the ability of the microbe to invade and multiply. And before we even get to the phase of invading our body, we want to do things to prevent it from entering our body through a portal of entry. So we could do things like get vaccinations, wash our hands really well, handle our foods safely, and clean and disinfect surfaces. And so by doing many steps like that, we are kind of creating walls or barriers for the microbe to enter in. The other term I didn't mention just yet is immunogenicity. This is the ability to induce an immune response. And sometimes this has to do with the viral load or the dose of the infectious agent within our body to trigger that immune response. Continuing our factors of infection, each microorganism has a mechanism of action, basically talking about what processes or factors does it use to damage our own tissues. Here's that term pathogenicity again. It's the ability of the agent in order to produce disease. And to do this, we are going to take into account our virulence factors, which I'm going to talk about more in the next slide. But essentially, we're going to see that the more virulent a pathogen is, the greater pathogenicity that it has. And we have two indicators of virulence. We've got the median infectious dose, which is the number of pathogen cells or the virions that are required to cause an active infection in about 50% of inoculated animals. And we have a median lethal dose, a number of pathogenic cells, virions, or a toxin that is required to kill 50% of infected animals. These terms, again, are review from micro. And then we've got our portal of entries where the microbes are going to enter into the body. We can look at this image here to see many of them as reminders. Our eyes, nose, mouth. We've got the placenta if the person is pregnant, the vagina, the urethra for both individuals here, and the anus, any broken skin that we have within our body, needle injections, and insect bites. And then we have toxinogicity. This is the ability of a pathogen to produce toxins to cause damage to our host cells. And we can divide this out as being an endotoxin or an exotoxin. But essentially, they're biological poisons that are going to assist in the ability to invade and cause damage in our tissues. Now, I mentioned I'm going to talk about virulence a bit more on this slide. Again, it's the capacity to cause severe disease. So we talked about in micro how we have many different types of virulence factors. In this case, it is going to be a capsule. This capsule is used in adhesion and aids immune evasion. I use the example of pathogens that have this as our phagocytes trying to hold on to a slippery bar of soap. It just helps it escape that immune response. Of course, there's a bunch of other ones. It could be a flagella, it could be iron-binding proteins or pili, and many others. But just so we understand what virulence is, it increases the pathogenicity of that microorganism. Now let's talk about the stages of infection. Again, this should be review. We've got the incubation period. This is going to occur in an acute disease after that initial entry of the pathogen into our body. The pathogen then is going to start multiplying in our body, and we start out with low numbers, so we aren't really going to trigger our immune response right away, so we shouldn't see any signs or symptoms here. And this can vary in timing depending on what the pathogen is. Even viruses are going to have different timing for this incubation period. And then we've got our prodromal stage. And this is when the pathogen continues to multiply. You could see our numbers are rising here, and they are going to start showing our signs and symptoms at this point because we have activated an immune response. So you might see things like a fever or pain or swelling, inflammation, nausea, diarrhea, and so forth. Then we have our invasion or acute illness period. You could see that taking place over here. Our signs and symptoms are going to be the most obvious in this stage and severe. The pathogens, you could see the slope has kind of changed and they're multiplying really rapidly and invading our cells even further. Then we have our period of decline, hopefully, right? We're going to start to see that that pathogen is beginning to decrease in number. Signs and symptoms at this point should also decline, and the patient may be susceptible to developing a secondary infection because at this point our immune system is still weak. We can then lead into a period of convalescence. This is when our patient is going to return to normal functioning. We may have some permanent damage from the disease that can't be repaired due to the high level of inflammation that may have taken. Of course, this is something that we want to happen, is to return to normal functioning, but the alternate to this is the disease being fatal or entering a latency phase. Now let's go through some of our microorganisms to review their properties and how they're going to infect, starting with bacterial disease. This is going to be a common cause for disease. If you remember, they are known as prokaryotes, meaning they do not have a nucleus. They can be aerobic or anaerobic. So if you remember, we've got these different classifications based on if they need oxygen or not. Aerobic means that they do need oxygen, so obligate anaerobes absolutely need it, whereas obligate anaerobes need to be in an area without oxygen in order to thrive. And then we've got our facultative anaerobes, in which it doesn't matter as much, and our aerotolerant anaerobes, which can be exposed to oxygen. It doesn't bother them as much. They can be motile or immotile, and of course we have different shapes of bacteria, and we've got classifications into gram-positive and gram-negative bacteria. If you remember, gram-positive bacteria have that thick peptidoglycan wall, or I shouldn't say wall because we do have a cell wall as well, that it is contained within, but it is a molecule that is created. And then with our gram-negative, we are going to have a thin layer of peptidoglycan and in addition of an outer membrane in which LPS is attached to. Remember, lipopolysaccharides are going to be a virulence factor. And our example is going to be our Staphylococcus aureus that can cause a variety of infections in different organ systems. Speaking of virulence factors, here is a list of some of them to review. We've got our pili or fimbria that really help with attachment or also can transmit genetic information to another bacterium. We've got flagellum that allow bacteria to travel, capsules we already talked about, enzymes that are going to help break down various proteins or other molecules, and competition for iron and nutrients, and lastly, our toxins. Let's talk about toxins a bit more in this next slide here. So first up, we've got our exotoxins. These are going to be basically our protein molecules that are produced by pathogenic bacteria. Majority of them will be produced through our gram-positive bacteria. They'll have more specific actions, so they really target certain receptors or certain cells so they can cause damage to those areas. Another thing that's unique about them is they're able to withstand really high temperatures, but will denature if they are in an environment where temperatures are above 106 degrees Fahrenheit. They're also very potent, so if we have a small concentration of an exotoxin, it can be lethal. And then we have our endotoxins. These are known to activate inflammatory responses and produce fevers. The example we talked about already is LPS and how they're able to do that through our gram-negative bacteria. And we know that bacteria can escape from our tissues and move into our blood. So when we have bacteria in our blood, that is known as bacteremia. And then if they start multiplying within our blood, then it is septicemia. Our gram-negative bacteria in our blood will also in the blood start to release many toxins. And that can cause a overproduction of pro-inflammatory cytokines, causing fever, activating our platelets to cause clotting. It promotes intravascular coagulation and permits escape of a lot of the plasma into the surrounding tissues. That can lead to a drop within our blood pressure that can lead to shock and eventually multi-organ failure and eventually death. Now let's talk about viral disease. This is a common affliction to humans. There are many ways in which they can be transmitted: aerosol, infected blood, sexually transmitted, animal reservoirs, and through vectors. So we know that viruses depend on the host cell for replication. They hijack our cellular machinery in order to make this happen. They cannot do it outside of our cells. So you could see the virus is entering into our cell here. First, we've got binding, attachment, adhesion. Depending on the source, they use different terms, but essentially you know that that virus is attaching to a receptor on the cell membrane. Next, we've got penetration. So it actually moves inside the cell. We can uncode it in order to release the genetic material and move it into the nucleus. At that point, the goal is to replicate that genetic information so that a ribosome can read it and create many viral proteins and parts. We can reassemble everything and then allow it to be released or bud out of our human cell through exocytosis so it can go and invade other human cells. What are the effects of viruses on an infected cell? It could be inhibition of that host cell DNA or RNA or protein synthesis. It can also disrupt our lysosomal membranes that release enzymes that can then cause damage to our host cell. It could fuse our host cells. You can kind of see that taking place over here, where cells have fused together and we have many nuclei and we create a giant cell. We can also cause alterations in antigenic properties, which then may also trigger our immune response to kill our own cells. Transformation of our host cells into cancer cells, in which hopefully our immune system will attack and get rid of, mainly our NK cells or our killer T cells. And it can also promote secondary bacterial infection. Next, let's talk about antigenic variation. We mentioned in the previous slide that we can alter surface proteins so the pathogen is not recognized by our immune system. And our antibodies are unable to recognize this, and it can lead to a chronic disease. If we have a minor change, this is called antigenic drift, in which we have point mutations taking place in our genetic reading, causing just a slight change in the spike proteins of our H and N. So I think I have this in an image down over here. Yep, our antigenic drift, where you could see our neuraminidase and our hemagglutinin, and those are the spikes sticking out of the virus here. And with this minor change, we start to see that, let's say, the H's are mutated. So just a slight change here. Then we have the process of more of a major change, which is an antigenic shift. This is a major change within the spike proteins due to a whole gene resortment. And you can see that that takes place when we are mixing the two genetic information into a new progeny. And this can take place when we have different species genetic information crossing over and both moving into a host cell and resorting that genetic information and creating basically almost like a whole new virus. So first question here, which virus undergoes yearly antigenic drift, allowing for emergence of new strains? Is it A, influenza, B, streptococcus, C, staphylococcus, or D, herpes simplex? If you knew it was influenza, you are correct. It goes through yearly antigenic drift resulting from mutations in their key surface antigens, the H and the N that we saw in the previous slide, and we get a new strain of the influenza virus. All right, now let's talk about fungal infections. Now, when we look at the structure, it is going to be a large eukaryotic microorganism. We see thick, rigid cell walls without peptidoglycan. Remember, we're going to find that within bacterial cells. And we see resistant penicillin and cephalosporins. So our single-celled yeasts are going to be mostly these spheres that we see, and they are facultative anaerobes. Our multicellular molds, which you could see pictured over here, will be structured more through these filaments or hyphae, and these are anaerobic, meaning that they are going to want oxygen. Their process of reproduction is through just simple division or budding. Now, if we have a disease caused by fungi, we call this group mycoses, and they could be superficial, deep, or opportunistic. Other mycoses are going to serve, or other fungi, I should say, are going to serve more as decomposers, or we can produce certain foods like cheeses, and they can serve as a source for antibiotics. Now, one group of fungi that are pathogenic are known as dermatophytes, and this is going to include our trichophyton, microsporum, and epidermophyton, if you remember from micro. So this group will invade our skin, hair, and nails. And so when you see dermato, I want you to think of our integumentary system, and phytes, just think of a fungi. So diseases that they cause are called tinnias, also known as ringworm, and we have the various different types pictured over here. We've talked about them before, so this should be review. For the pathogenicity, they are going to adapt to their host environment. So they're able to handle a wide variation within temperatures. They can digest keratin within our skin, and they're able to survive in low oxygen environments. Usually we can control them through our phagocytes and our T lymphocytes. And I put this picture in here just to kind of show how they are able to kind of go through our keratin and our skin and establish themselves and continue to grow their hyphae throughout our skin. Here we have our fungal infection that is most well known, our Candida albicans, and this is going to be an opportunistic yeast. We do have this in several areas of our body, and it's known to have a commensal inhabitant of our normal microbiome in areas such as our skin, GI tract, and the vagina. We see that we can have a localized infection where an overgrowth occurs. We can have thrush within the oral cavity that we've seen or talked about in our previous lectures, or within the vaginal canal like you see pictured here. Disseminated infection can take place if we are immunocompromised. So we could develop a deep infection, and that can lead to high mortality rates. Now let's talk about parasitic infections. This is symbiotic in nature. It's going to have a large size variation, so we can have anywhere from a unicellular protozoa, such as malaria, amoebae, or flagellates, or they could be really large, like helminths known as our flukes, nematodes, and tapeworms. This is transmitted through vectors or some type of ingestion, and they can cause tissue damage that is secondary to infestation itself with toxin damage, or from inflammatory or immune responses. And I just wanted to mention this is a little outside of the range of what we're talking about, but how we can break the chain of transmission. And it depends on, of course, what type of microbe we're trying to block, but these are just worth reviewing as a reminder. Now, what about antibiotics? They help to prevent the spread of infection. There are antibiotics that are bactericidal that will kill the microorganism, and others that are bacteriostatic that help inhibit the growth. Now, we've got a lot of different types of antibiotics. I'm sure you remember memorizing them in micro. And they work through different mechanisms of action. What they could do is work to inhibit the function or the production of the cell wall or cell membrane. So we've got the cell wall pictured over here. Prevention of protein synthesis through the ribosomes. We can block DNA replication, and we can interfere with folic acid metabolism, which is important to bacteria. Now, sometimes we have certain bacteria that form antibiotic resistance, and this can result in death. Resistance to a single antibiotic has progressed to multiple antibiotic resistance. And remember, this can happen by somebody maybe not taking their full recommended amount of antibiotics. So what it ends up doing is killing off most of the bacteria within the body, but then we can keep certain bacteria that have been mutated and are able to resist that antibiotic, and then they can continue to reproduce, and now we've got a lot more bacteria that are resistant and more difficult to treat, so we can try switching antibiotics that could be a little bit stronger in order to combat them. This can also take place if we have an overuse of antibiotics. Next, we've got our vaccines. These are biological preparations of antigens that stimulate production of protective antibodies. It could be a weakened microbe or a killed microbe that is put into the vaccine, and this usually is going to provide long-lasting immunity. This is going to trigger our primary immune response that is short-lived, so sometimes we do need booster injections in order to push our immune response to secondary responses so that we can create a large number of memory T and B cells that are prepared when we come into contact with that pathogen for a second or third time. And so we can create protective levels of antibodies or our T cells in this way. The CDC does have schedules for child, adolescent, and adult vaccinations. I believe you're going to cover this in another course, and we briefly discussed it in micro. Lastly, we're going to talk about passive immunotherapy. These are preformed antibodies that are given. So we have a human immunoglobulin for people with hepatitis, immunoglobulin and monoclonal antibodies for rabies infections, and monoclonal antibodies for RSV.